Okay, today is day 2 of the bioinformatics class. I'm really glad you all came back. We are having an interesting discussion today because we're going to delve into this world of imaging informatics. Imaging informatics is a fascinating place with an awful lot of really cool advances, and it's all going to build upon what we learned yesterday about how to represent visual information in bytes. So we're going to start with a little discussion of fluorescence microscopy and move into flow cytometry, since these two have a lot in common between themselves. So we're going to start by measurements of light intensity and figure out how we go about making these measurements. How do you turn a visual signal into something that you can store on a computer? We're going to talk about the problems of processing image data with the emphasis on microscopy at first, but then we'll shift into flow cytometry. When I came to Stellenbosch University, I joined an immunology group, and I have to say they use a lot of Luminex assays and fluorescence, cell sorting and things like that. So these are technologies we should know about. I know that they're not as heavily emphasized over here, but I think that understanding flow cytometry is very valuable. Okay. So if you spent time looking through a microscope in your cell biology class as an undergraduate, you probably had an instrument rather like the one on the left. We have the sample down at the bottom. We have some sort of light source that's sending light to it. Light is reflecting from that or is being transmitted through the sample, depending how thin it is. It hits an objective lens, passes through some other optics. It comes to an ocular lens, and that ocular lens is closest to your eye, exactly, and your eye then serves as a detector. So we have a light source, we have reflected or transmitted light that is then passing through optics being detected by your eye. This all gets turned on its head a little bit as we go to fluorescence microscopy instead. So in this case, we have light that is rather special. We're using laser light to light the sample. That laser light is different from visible light in that it's monochromatic, it's one frequency, and it's polarized, and it's a laser, right? So this laser will pass through the various lenses to get to the sample. That light is taken up by fluorescent dyes. They become excited and they emit light, and that light is emitted and passed out to a PMT, a photomultiplier tube. So instead of having a lamp serve as your light source, you need a laser because you're trying to emit particular frequencies. You have dyes in the sample that are able to emit light in response to that laser, and you're not detecting by eye, you're detecting by a PMT. So that is a very brisk overview, but talking about PMTs is also worthwhile. A photomultiplier is something that we can make very, very small. I know that this looks like a can with a whole bunch of mirrors in it and so on. But in fact, PMTs can be made very, very small. And as we mentioned yesterday, sometimes the cameras we use in our phone have millions of these things packed into the surface in a charge-coupled device. So these PMTs, the photomultipliers, are very, very tiny to make and are exquisitely sensitive. So here we have an incoming photon at the left. A photon is a particle of what? Light. A particle of light. All right, and so the photon hitting this first electrode kicks loose a shower of electrons. Those electrons then pass to the next electrode, and their arrival at this next electrode causes an even bigger shower of electrons. That shower of electrons hits the next electrode, and boom, it's amplified again. So what we have is this huge step up in strength. What arrived was an individual photon. What was emitted is a shower of electrons. And by creating this shower of electrons, we create a current large enough for measurement. Okay, so a PMT is a way of turning light into a shower of electrons that can be measured as a current. I gave myself a whole lot of slides today, so I may rocket through these to some extent. I'm trying to touch on anything that would actually be relevant on a quiz, for example. All right, I think most people remember the electromagnetic spectrum, but just to test people a little bit, I have red light at one end of our visual spectrum, and I have purple light at the other end. which one is more energetic? The purples. The purples are more energetic, that's right. So as we get to low wavelengths, which way are frequencies going? Right, right. So as you go to a lower wavelength, the waves are shorter, thus you can pack more of them, and so you end up with a higher frequency, you have more energy. So this is the energetic end of the spectrum, this is the less energetic end of the spectrum. Correspondingly, when we look at the kinds of waves that show up at the large wavelength end of the spectrum, things like long radio waves we don't think of as energetic at all. I mean, you might occasionally see a post on Facebook about how I live near a power line and it fried my baby and so on. And it's terrible that awful things happen, but in fact, power lines didn't do it. So we see that we've got like long radio waves, AM and FM, microwave rays, IR. IR stands for what? Infrared, very nice, good. And then we get to the stuff that's more energetic than visible light. UV, which stands for? Ultraviolet. X-rays, I think everyone accepts that those are pretty energetic, right? There's a reason why your dentist puts on that lead apron every time you show up for visualization. And then we have a little symbol with rays by it. Anyone know what that symbol was?
Gamma.
Gamma rays, right. So if you want to make the Hulk out of Bruce Banner, you need that. Lots of energy. I accept that the Hulk was not created, that's fine. On we go. So, remembering frequency and wavelength matters to understanding how this light works. So, fluorescence frequently has to do with these chemical compounds with rings in them. So when you have aromatic rings like this, there's some likelihood that you have the ability to absorb a particular frequency of light and emit a frequency of light. In natural proteins, what amino acid accepts light and fluoresces in response? Phenylalanine, okay. Are any others? Tryptophan, right. So some of the amino acids we've got have these double ring structures in them, and it causes them to fluoresce under certain circumstances. In some technologies, we'll see that autofluorescence is used to detect the amount of protein that happens to be floating by at a given moment. Okay. Now, when we think of lasers, probably the most frequent kind that you encounter is the laser pointer, right? You've got a couple AA batteries in there, and then they've got this little dude that's taking that voltage and turning it into a laser beam. But I would note that lots of us still have DVD players and Blu-ray players, and for that matter, CD players, and all of them make use of lasers as well. And the structure, the size of the laser in these devices is vanishingly small. So here we have an American penny, which is not all that different than the 10th of the rand piece, and we see just how small these little lasers can be. We can get lasers that use all kinds of different chemistries, and these different chemistries result in different different frequency outputs for those lasers. Does everyone remember the term Stokes Shift from undergraduate chemistry? Oh, good. Well, we'll have a moment to remember this. All right, so we know that mass energy are neither created nor destroyed. So in this case, we are accepting some light at the fluorophore, and we're releasing some light. We do have some amount of inefficiency in this transfer. As a result, if you have an input wavelength of 495 for a fluorescein molecule, we see that the light it emits is not at the same frequency as what it's accepting. Instead, we see a shift to a longer wavelength. Is that a shift to higher energy or lower energy? Lower. So there's a little bit of inefficiency involved in this lighting process and the emission process. We put in more energetic light and receive back a less energetic light, one photon in, one photon out sort of thing. That difference between these two frequencies is called the Stokes shift. I'm sorry, I talk about frequencies and wavelengths kind of interchangeably, but remember that one has an inverse relationship to the other. As we go to a higher wavelength, we go to a lower frequency. Okay. Now, the situation becomes a whole lot messier when we're thinking about multiple dyes in the same sample. This is a theme that's going to show up pretty strongly in a concept that we call compensation. Okay, so remember our friend Fluorescence, he's right here again. So we are now looking at the fluorescent intensity of the emission spectrum, I believe. So we're looking at the light that comes, that is received back from these, and we see that when we look at multiple dyes in operation within the same sample, the emission bandwidth of the emission spectrum of one may overlap with the emission spectrum of the other. That's a little messy, because now you could be receiving only this red signal and think that you're seeing a bit of the blue signal just because their spectra overlap. So that's problematic for us. We want to be able to translate the amount of light we receive into a quantity, or to say there are some number of fluorescent bodies in the sample and it's this. But if you have crosstalk going on, it can be a problematic situation. Do any of you have a sibling who's talkative? Sometimes it's difficult for the quiet one to get a word in edgewise if the other is speaking volubly. I'm just saying. Not that I have an older brother. Hi, Tom. Nice to see you. So, anyway. All right. So in the same way, when we have multiple fluorescent dyes that are all communicating in the same sample, it's possible that one may mask the other because they have some overlap in their fluorescence emissions. Now, contrast is a big deal. I think folks are going to remember the image of a mite head that we were looking at yesterday. We looked at it in 2-bit, just black and white, in 1-bit, black and white, 2-bit, 2 shades of gray plus black and white, and so on. Contrast, it gives us the ability to make out the details in these images. And the way in which the visual details spill out across the range of intensities that are available can make a big difference in our ability to retrieve information from it. So dynamic range is a comparison between the brightest and darkest return that you get from an image. If you have 16-bit brightnesses, very high resolution brightness distinctions, and you're only working within the top 4 bits, you're losing most of your ability to discriminate a high signal from a low. You don't have a lot of contrast, in other words. So having the light return from a sample be well within the range of intensities you can actually capture makes a very big difference. Optimizing contrast facilitates our ability to get information out of microscopy data. And I'm going to try to walk you through a quick example of that. So let us imagine that we have captured this image on our microscope. We've all seen something like that, right? We took a microscopy class somewhere along the way. I'm not a good microscopist. But imagine I have produced this image. Now, can I get my data out of it? Well, let's start with a histogram. Everyone remembers a histogram, right? We produced one of those yesterday. In this case, we're going to ask, what is the popularity of each level of intensity across the multiple megabits of this, across the megapixels that we have available to us. Maybe we've shot a 10 megapixel image. That means we have 10 million intensities, and we want to see which ones are most popular. So we see that in this case, we have a whole bunch of measurements that fall in this medium range intensity, and some that spread out to the other ends. Now, have we used the very darkest colors in this image? Not really. So black is all the way down here, and we see that there's really no appreciable number of pixels with total black to be seen here. We did, however, use some values all the way up here at the white end. So we might have, one way that you could read this is that you've overexposed it, but that's not really the case because we don't have, say, this edge butting up against the top. So, but we certainly haven't used the very bottom of our intensity range. So if we're trying to interpret these data, these cell images, we're going to run into this problem that pretty much everything is kind of muddy right there in the middle. So one of the things that we can do is to alter what our input brightness, how our input brightness maps to an output brightness. We're going to apply a brightness and contrast filter to it that is going to allow us to change which brightnesses get mapped to what. And so if we flip back and forth between the first image and the right, we see that we got brighter But we still have this problem that there's not a lot of contrast. We don't have a lot of details that are showing up nice and dark against this bright background. So this isn't really an optimal color curve for interpreting this image. If we alter it again, we can alter it so that this mapping function of how the input brightness becomes an output brightness is really maximized across where most of the values appear. So we see that we're not going to require something be black in the original image in order to be black in the output image. We're just saying that the darkest image, the darkest pixels will now be called black, even though they were only dark gray to begin with. Now the result is that suddenly we have a much better contrast in the image that we see. You can imagine that if you were to take a family photograph on a rainy day like today, you might have an awful lot of gray and muddiness in the picture. And if you want things to stand out better, if you want faces, the people to stand out against the background, you can change this brightness and contrast function so that that's optimized. In this case, by using the histogram of intensities that we originally collected, we can figure out where the informative content is, set up our brightness and curve function, and now a much easier to interpret image has resulted. So we may have started with this, we tried brightening it to begin with, but then found that if we optimized our color function, our mapping function between input and output brightnesses, we got a lot better contrast out. Contrast is how you get information out of biological imagery. Okay, now let's talk a little bit about kernel convolution. Kernel convolution is a very widely used technique. I imagine that many of us have taken a poor photograph at some point in our lives, and we tried to clean it up in Photoshop or Paint.net or whatever tool you happen to use. Anyone have a favorite software for touching up photos? A few? Okay, I appreciate that we could call something like Instagram, if you want to put a bunny nose on your face or whatever, that you could consider that improving the photograph, I guess. But in this case, we're talking about convolution filters, and that's not the kind of thing we're typically thinking of for Instagram. So one of the things that we frequently use is like a smooth filter, or an edge detection filter. So today we're going to understand how those work. In convolution, we are making use of multiple pixels in the original image to produce one pixel in the output image. So again, we have a raw image coming in, and we're going to dump out another image that's been modified in some way. So we have a convolution filter, in this case one called a Sobel, that is, in this case, a 3x3. So it's got 3 dots across and 3 dots down. So we've got 9 pixels in each source image that will contribute their value towards making 1 pixel in the output image. In this case, we've drawn the convolution filter at the upper left-hand corner of the input image. So the first 3x3 grid in the input image is being used to create the second row, second column dot in the output image. Everyone sees that? Okay, but how do we combine multiple input values to create one output value? Now, which filter you're using has everything to do with what's going to result. So we're going to start with the Gaussian kernel filter. The Gaussian kernel filter. When I say Gaussian to you, what's the first image that comes to mind?
Black.
A what?
Black image, nothing.
Oh, nothing at all comes to mind. That's okay. Okay, a Gaussian distribution. How about a Gaussian distribution? Anyone had statistics recently? How about a bell curve? Yeah. Bell curve, great. A bell curve is the Gaussian distribution. Okay. So a Gaussian distribution is one of these bell curves. And in this case, a Gaussian kernel filter means that we're going to have a two-dimensional Gaussian distribution. So not just like this and like this, but also like that and like that. Right? So in both dimensions. So the Gaussian kernel filter is going to grab information from a grid of 3x3 pixels in the original image and use them in a Gaussian kind of way to combine information across those 9 points. So in this case, you can see the weights that are being placed on each of those 9 points. Which of the 9 points gets the strongest weight in the output?
4.
4. And that one is located where?
Center.
In the center. Okay, so that is to say that the point in the input image that is located at the same location as the output in the output image is going to have the greatest weight in determining what that color, that brightness will be. But it also places weight on the surrounding ones. So things that are directly north or south or east or west, sorry, here is east and west, those points are going to give half as much weight as the center for contributing the value of the output. And, northeast and northwest and southwest and southeast are going to get a little bit of weight in the output. So when we total that all up, we get 4 points, a weight of 4 for the top row, a weight of 8 for the second row, and a weight of 4 for the last, for a total weight of 16. So we're going to multiply the intensities that we had on our input, add them all up in this way, and then divide them by 16 to figure out what the output will be. All right. So I'm going to skip past this now. So those inputs and outputs can have a really dramatic effect. So Gaussian filters are really, really good when you have a noisy image. Imagine if you had just this tiny little bit of snow in the picture, right? That's going to obscure the details that might be within it. And so in image processing, we would consider that high-frequency noise. It's just a speckle that appears in one pixel but doesn't affect the values around it. And if you use a Gaussian filter, you're able to resist those snowy features very strongly to get rid of it. And so here we have an input, here we have an output, and that Gaussian filter has done a little bit of cleanup. I don't actually think it's very, very clear in this particular example, but the amount of high-frequency noise, the little speckliness of the image has been diminished by the use of a Gaussian convolution filter. But this is not the only convolution filter. There are lots of different contexts in which we can weight values that are above or below or left or right of a pixel to figure out what we really want its value to be. So this one is called a Sobel filter. This is the example that was appearing in the very first slide we used. Now you can see that its weights are quite different. It's going to completely ignore the intensity on this row, but it's going to use the values above and below in contrary ways. An upweight on the values above this and a downweight on the values below this. And you can see the image that comes out is quite weird, right? But look how strongly this has drawn out the intensities of horizontal features in this figure, right? You could turn the Sobel filter on its side and get vertical features. Here we can see that it's really strongly picking up horizontal edges from the original image. Remember, the point is not to make something that's photographically pure in some way. The point is to maximize our information from these images. So if your goal is to find boundaries of things, using a Sobel filter is a really good option because it's going to highlight those very strongly. It's quite different than the purpose of a Gaussian filter, which is designed to make the noise go away. Here we're not talking about the original content of the image much at all, except that where we've got these horizontal crossings, we want to make them light up very brightly. Okay, so I have a whole bunch of vocabulary words on a slide. I always try to kind of broadcast the slides that I think are most valuable for assessment points, and this is one. Any term that I put in italics on this slide is likely to be something that I want you to have in your vocabulary list. Okay, so in image analysis, we frequently talk about segmentation, which is the ability to separate objects and background from their visual field. Just the same as when you're watching the video, I'm hopeful that you're noticing motion and animation in the teacher and probably ignoring whether people are walking by in the background, right? I want to be the star of my own video. So, registration. Registration is something that our brains have to do all the time. We need to be able to align two images of the same scene by mapping objects seen in both. Who has gone to see a movie recently? Anyone go see Aquaman? Oh, a few people saw Aquaman, that's good. Someone thought it was good? Ah, good, good. We have some enthusiastic DC fans here. That's good. All right. How frequently, when you go see a movie, is the screen updating? Is it continuous action? To some approximation it is. Our brains make us think it is. But in fact, the picture that you see on screen is changing every 24th of a second. It's playing at 24 frames a second. Now our brains are able to let those images persist and melt together enough that you don't have a lot of difficulty saying, in this first frame I saw Aquaman going like this, and in the next moment I saw him move his hand very slightly, right? It's the 24th of a second. He's not going to move very much, even though it's very fast. So this is the ability to register that a hand in one frame maps to the hand in the next frame. In biology, we need to do this all the time. Imagine if you were to take microscopy data from cells in culture, and you're taking a new snapshot every minute over the space of a day. Your ability to recognize that this cellular boundary has moved, maybe you're looking at amoeba, I don't know, and this pseudopod has grown slightly in this direction, requires that your brain be able to say, or that the computer be able to say, this pseudopod is the same as that pseudopod, and this point maps to that point between these two images. That is an exercise in registration. And that relates very strongly to motion analysis. If you're trying to make your image information correspond to time, to say that this particular blood cell has moved through circulation at this many meters a second, you're trying to answer a motion analysis question. And of course, quantitation. Being able to turn something like, I saw this amoeba move over here, and then over here, and then over there, and over there. How many microns has this amoeba moved in this 20-minute interval? Right? That's difficult because now you've got to deal with the fact he's been moving all over higgledy-piggledy, just all over the place. And so now being able to say, what is the total linear distance that he's traveled over what period of time is a quantitation question. So these things are hard to do, and yet we've got really good tools to support stuff like this. So in segmentation, I told the story of these before I actually got to the individual slides. I'm sorry about that. For segmentation, we can imagine that in this case we have cell nuclei that we've stained with hooks. You can probably pronounce that better than I. And we have them in this field, and now we need to be able to differentiate these nuclei from their backgrounds. You can see that when you have a cell way down here that's not next to any others, drawing a little red boundary around it is not a problem. But when you have a whole bunch of these nuclei all bunched together in one place, now you have overlaps that are a lot harder to deal with. So this is an example of why segmentation can be very hard. If you look at the slide on your laptops, you'll see that the software has drawn in little red boundaries on each of these nuclei as well. It maybe is not as clear here. So remember, that's kind of the positive and negative thing. How can we pick out an object versus the background in front of which it sits? All right. Now, 3D reconstitution is a very big example of the registration problem. So what do I mean when I say a Z-stack or a Z-stack? In this case, I have my individual fluorescence microscopy images, but I've altered how deep within the sample I'm focusing, which means that by changing how deeply I'm looking down into the sample, my images are able to capture basically thin sections of the transparent material within it, so that I can say, this cell has a volume, not just a cross-sectional area, but a volume of so much. Or this network of microtubules has this kind of connectedness. So this problem of 3D reconstruction is something that certainly requires registration very strongly. Now, I am very fond of videos, but I have to say I don't include them in slides very often. So if you look at this YouTube link on your own computer, you'll be able to see an example of this motion analysis. Here we're looking at cells emerging from the embryonic quail heart. You see all of these little traces? That is a computer reconstruction. This is not the image data themselves, but rather the software being able to track how individual cells are moving from frame to frame over an interval of time. So there's a world of really fascinating videos out here that have been produced by people in fluorescence microscopy. I remember that one video I saw from a friend of mine who presented this class at Vanderbilt was to look at the motion of red blood cells in a network of blood vessels near the heart. It was astonishing to see just how quickly these little dudes go flying around, and trying to quantify the rate at which they're tumbling through the blood cells was astonishing. Okay, and of course we love quantitation. Now I spent 10 years doing colon cancer research back at my old place. So when we do quantitation, we may be trying to ask about the spread of a cancer. What is the tumor volume as a function of time? So we can do this by using our fluorescence signal and trying to quantify from it. So quantitation is a big deal. If you have a pretty picture, that's great, and it'll probably play well in a conference. But if you're trying to pull in quantitative information from it, having the ability to do this bioinformatically is going to make a big difference. I've mentioned that we have some tools available for this, and ImageJ is a really important one to know about. You might think you have to spend a whole lot of money to do bioinformatics on imaging data. That's not actually true. ImageJ is free software. You can just download it directly from NIH, and it can do some awfully useful things. Everyone heard of C. elegans? C. elegans? Okay, it's a worm that is very, very favored by the developmental biology community. So here we have a worm. If you are trying to evaluate the perimeter of this worm, you could sit there and lay a string on the print of the photograph and then figure out how long a string is its circumference. Or you could just use a plugin within ImageJ for detecting that boundary of the worm and give you the linear measurement of its circumference for free. So that's a pretty good thing. There are a great many plugins available. So if ImageJ doesn't do what you want it to do out of the box, you can just download a plugin in many cases that will handle that task for you. All right. Now, before we just blow right ahead, I want to stop and see, are there any questions about things like registration and segmentation, motion analysis, and quantitation? Are those pretty clear? Is the operation of convolution filters sensible? Does that ring a bell? Remember we talked about those? People remember that. Okay, but maybe a little less comfortable. Okay. Do we understand why maximizing the contrast between bright and dark features in an image is necessary to getting the best information from it? Good. Okay. Well, I got a raised eyebrow on that one. That wasn't so great. So maybe even that point could be made better. I'll try. Okay. Now, flow cytometry puts us in a rather different place. We want to be able to enumerate particles in suspension, which is kind of a dry way to put it. Frequently, what we're trying to do is count cells of particular types. That's really what the magic is. So along the way, we're going to need to discriminate between biological and non-biological phenomena. If you're trying to count cells and you mistake other components in your sample as cells, that's going to be problematic. Frequently, we want to be able to differentiate live and dead particles, cells in this case. We want to be able to evaluate a great many of them. Here I've said 10 to the 5th. What's 10 to the 5th? 100,000. It's 100,000. 10 to the 6th is 1,000,000. 10 to the 5th is 100,000. So if you have 100,000 cells in suspension, and you want to be able to evaluate parameters of each of them, flow cytometry is very powerful for that purpose. We want to be able to measure particle scatter as well as innate fluorescence. We already mentioned an example of innate fluorescence for proteins, because when we have double ring structures in our side chains, it can lead to the fluorescence of those proteins. We may want to measure secondary fluorescence, which is to say the fluorescence released when we illuminate the samples with a laser. And in some cases, we actually want to sort. We want to push all cells of a particular type into this vial and everything else into another. That too is possible within flow cytometry. Now, all of this comes from a guy named Coulter, who argued, cells are not particularly good conductors, right? If you pass water with a bunch of ions through a flow cytometer, that's going to pass a current quite well. But the more cells you pack into that volume, the worse its ability to transmit voltage, transmit current. So blood is a suspension of cells in plasma. Plasma is a relatively good conductor, but the cells are not. So in the past, people have done cell counting on blood by just passing a current through it and finding out what the conductance was. But as the ratio of cells increases, the conductance of blood decreases. So this is the Coulter effect. It's the principle on which flow cytometry works. Now, this is not that very different than what we were looking at just a moment ago with the fluorescence microscopy. We still have a laser. We're still lighting up cells that are passing by. So you can think of this as kind of a microfluidic system. We're trying to create droplets where each droplet is one cell passing through. This laser can illuminate that cell, and some amount of light will pass through to this photon multiplier, which is giving us the forward scatter measurement. Some of it is going to be reflected off of the cells, off in another direction. That may give us the side scatter measurement. We may also have other lasers shining on this that are lighting particular fluorescent dyes within the cell. And for that, we may have other photomultipliers that are recording those intensity values. And all of this has to be done in real time. We're not just parking a cell and analyzing it for several seconds, right? These are characterizations of cells as they pass by. So all of this can be analyzed by an analysis workstation that can do things like gate the cells to direct cells of a particular type into different cell collections. All right. Now, if you are using multiple colors, multiple lasers all shining on this sample to get these fluorescent activities out of them, you're probably going to run into this problem that we spoke about with the fluorescence microscopy, that the emission wavelengths of these different dyes can overlap. So if you have a lot of PE dye, a lot of emission coming from that, it could potentially mask this yellow dye, the PETR dye. So that would be problematic. We need to be able to adjust how much intensity we're seeing at these levels to pull out the contribution from PE. And that's a problem we're going to call compensation in a minute. Oh, here it is, yes. So if you imagine that we have these four dyes in operation, we need to be able to subtract out how much the yellow is contributing to our orange signal in order to be able to make an accurate quantitation of how much orange there really is. So in this case, I'm showing 4 dyes, but there are instruments that can measure far more dyes than just 4 in a single one of these. Being able to discriminate them from each other, being able to adjust for a very loud signal at a neighboring wavelength is something that we frequently must do. Okay, so gating is one of the most frequently used bioinformatic tricks in flow cytometry. But I'm going to try to make a distinction between real-time and after-the-fact a posteriori software gating. So in real-time or live gating, we're only going to record to disk certain events, the ones that meet our criteria. So imagine that you're trying to characterize each droplet that's passing through the flow cytometer. Some of them have 0 cells. Some of them have 1 cell. Some of them have 2 cells or more. We don't really want to store information about droplets that contain multiple cells because we have no guarantee that they're the same kind. We don't want to store information about the droplets that have no cells because they've got nothing to tell us about our biological story. So having a real-time or live gating procedure that rejects information from any cell that has, any droplet that has more than one cell or less than one cell is a very appropriate thing. On the other hand, we're going to use a lot of this gating to analyze the fluorescent responses that came from these individual events. So if we have completed a flow cytometry experiment, we can come back to look at what the fluorescent responses were for each of the dyes on each cell as it was passing through. And that's the example of software gating that we would talk about. So why bother with this at all? Frequently, we're using fluorescent dyes that are coupled to particular antibodies. So we're not measuring innate fluorescence here. We're measuring the fluorescence that we've added that are tagged to particular functionality of the cell. So in this case, we have SSC. I think we've seen that one before when we were looking at this. It was side scatter. And CD45. CD45 is a particular antigen on the surface of these cells. And it's been marked with Pacific blue. So we see that if we look at all of the different dots, remember there were 10 to the 5th, 100,000 different cells that passed by in this time. Each of these dots then represents one particular event. This one particular cell gave us a side scatter measurement of this, and it gave us a Pacific blue measurement of that. So here we see a little dot there, little dot there, little dot there. Some places, though, we've switched to a different color. That's because this is a 2D histogram, and it's using these other colors. The closer you get to red, the more popular that output ratio was. So we see that we have a huge number of events where we had very little Pacific blue coloring and very little side scatter coloring. These things are not interesting to us. They're basically non-responders. On the other hand, if we look at this ratio of Pacific blue to side scatter, we see that there's a large collection of things that fall into this general pattern, a pretty high Pacific blue measurement and a pretty high side scatter measurement. And we have two other collections of cells down here that score even higher than the granulocytes on Pacific blue, but lower on side scatter. And these divide into monocytes and lymphocytes. So if you are trying to parse apart all of these different cell components of a mixture, the flow cytometry data is giving you the ability to differentiate them, in many cases based on things that are specific to an antigen you really care about. So gating, in this case, is going to mean that the person using this software is going to specify rules that say anything that falls within this oval but not within that oval shall be called a lymphocyte. Anything that's found inside this oval but not that one can be called a monocyte. These rules are the gating strategy for interpreting flow cytometry data. Okay, so one of the most basic kinds of rules that you can lay down in two axes is called a quadrant analysis. In this case, we are looking at FCS on the x-axis and PE on the y-axis. In this case, we're using a threshold on both axes. We're saying anything that's greater than 10 to the first on FCS is a positive for FCS. Anything to the left of 10 to the first on FCS is a negative on that axis. Anything higher than 10 on the PE axis is a positive for PE. Anything below it is a minus. And you can see that when you apply a linear, a fixed cutoff on the x and a fixed cutoff on the y, you're separating all of these events into 4 pieces. That's why we call it quadrant analysis. So this is a pretty simple way to set up rules. It's not always feasible. I mean, you remember the previous slide, we wouldn't be able to separate the monocytes and the leukocytes, I guess, because they were quite close together. There's no single quadrant rule that cuts those apart. But in some cases, it's more than sufficient to help us to separate out our cell populations. Now, it gets even messier when we get into sequential gating. So imagine in this case that we have several different channels we've read. We've got CD11B, side scatter on channel A, B220, TCR beta, CD8, CD4, SCA1, and CKIT. Right? That's a lot of different channels to be measuring on these cells. But in this case, these guys are after a cell population that's that requires many markers for us to be able to distinguish. So in this first stage, we're going to separate from all myeloid cells, this particular population of them. Any cell that passes this rule can then be passed on to this second rule. In this case, we're going to pull out these cells for consideration down here on those two axes. These T cells, on the other hand, we're going to separate on CD8 and CD4. So that these are the CD4 positive T cells and these are the CD8 positive T cells. So we're going after 1, 2, 3 minority cell populations, plus these B cells, I guess, 4 different cell populations, by using a combination of different markers. So in this case, we're using multiple stages to figure out which particular cell population matters to us. I think it's become pretty clear already that the complexity that's possible when we measure lots of different markers for how we're going to do gating can be really kind of frustrating if you're the person sitting at the computer saying, okay, for sample number 200, we want to apply this rule, and then we're going to apply this rule on top of this population separated out from the first step. Doing this again and again and again is a very time-consuming process. Most of the tools that we use for doing this gating are relatively manually intensive. So in the end, it is really somebody working for a university who's saying how these rules shall be applied to each sample. Very time-consuming. But on the other hand, you can publish in Nature Biotechnology, so that's pretty good, right? Okay. Okay, now, is it possible for you to make use of flow cytometry? It is. Now, I don't know what the flow cytometry installations are like here at UWC, but I know that just across the rail yard, we have our very own facility from CAF that allows us to do flow cytometry. So I asked Andrea, our researcher in charge of this facility, if I could come by and have a photo day, and she was very kind. I didn't include a picture of her. I probably should have done that. I'm sorry. But I wanted to point out that we have 1, 2, 3, 4, 5, 6 different instruments that you can use. Some of them are very simple. If you're just trying to say, how many cells do I have per milliliter, that's a trivial kind of question to ask. If you're using fluorescence markers and you need to pack a whole bunch in there, that becomes a bit more complex, right? So this is the LSR2. This is currently our Cadillac. So you can measure 12 different colors of fluorescence on this instrument. That's pretty great if you're trying to pull out a very small subcell population out of a complex mixture. And that's good because I hang around with immunologists and I tell you they use this stuff. We also have things like the Canto 2, which can measure 8 channels. So these instruments are all available, and if your experiment really needs one, if you don't have one here, you can use ours. You just we'll have to figure out the price for doing those operations, but I'm sure that's not going to be a huge barrier. Now, as we measure more and more colors that we're trying to use, things get harder and harder for us to interpret a particular gating strategy. It's harder and harder for us to apply these rules, even if you have one canonical set of rules you want to use for gating across a very large number of samples. So we think of humans as being particularly good as long as we're dealing with 3 colors or less. But once you start getting to 4 and 5 colors, your ability to do this as a researcher is going to diminish. And you might have irregularities that creep in when you try to apply the same gating strategy to a very large number of samples. This is particularly awful if you happen to be working with Cytoph. Has anyone heard of Cytoph? It's still a relatively new technology, maybe the last 5-10 years. Many people have said, look, the more colors you try to jam through these flow cytometers, the worse the problem gets. So why don't we stop dealing with fluorescence as our readout and instead use mass charge and intensity as your readout? Mass spectrometry is a really great field for separating out a whole bunch of different signals that are all competing for this space because it can discern things with different mass to charge values very, very accurately. So in this case, the Cytoph is doing a mass spectrometry measurement of intensity instead of a fluorescence measurement of intensity. So our dyes now are not absorbing laser light and emitting fluorescence. Instead, we are marking our particular markers with different metals. Do folks remember EU? I think it's europium, isn't it? And we've got let me see, erbium and ytterbium, goodness me, barium. I've forgotten all the different names. These metal markers, when measured in a mass spectrometer, mean that instead of limiting ourselves to maybe 10 different fluorescent dyes, we're instead able to measure upwards of 20. I guess when I produced this poster, it was this slide, it was already possible to measure 30 different 30 different markers within these cell populations. And you can mark external cell things, so proteins that appear on the cell surface can be labeled. You can even do internal labeling of marker molecules that are within the cells. So all of these can be differentiated from each other very cleanly by CyTOF instead. Now, CyTOF is not available at my campus, but the folks over at Quadrant at UCT Med School do have a CyTOF in operation. So this technology is available here as well. Before we wrap up, I think it's really important to remember some practical realities, and one of these is the FCS file. Frequently, when you're working with flow cytometry data, it's going to be made available to you in FCS format. Even if you are, if you're working in a particular field where people have previously published flow cytometry data sets, they'll frequently make those available to you through repositories. So you can download FCS files from somebody else who has done a related experiment and reanalyze those as part of your own. Kind of getting free experimental data. There's a lot of opportunity for that. In fact, I think our lecture, is it tomorrow, is about databases and repositories. So we'll be talking about this topic quite a lot more. The FCS file has been around for quite a long time. Now, I believe that in this room I am the only person who was alive in 1984. Am I right? I am right. See, I'm an old man. I have memories from 1984. That's even creepier. So FCS was created all the way back then, and I was born in 73. Thank you very much. 73. I'm a 45-year-old man. All right. So back in 1984, this field was already coming to the consensus that we need to have a standard way that we can communicate flow cytometry data to each other. And so they created the first file format for this in 1984. 6 years later, I was in high school, I was learning to drive. A very bad driver when I first started, I have to say. They were already updating this format to its version 2. Version 3 was introduced after I entered grad school in 1997. And these days they're up to version 3.1, and they've done quite a lot to make these files usable in different continents, on being able to store how compensation should be conducted, how we should visualize these data by default. Metadata, remember metadata? If you'd like to store what part of the experiment this particular file relates to. What about data provenance? Provenance. It's a lovely word, isn't it? Provenance. Provenance describes how you got it. Kind of the chain of custody is another way to think about it. What did the sample go through to get us to here? How have you analyzed the data over time and written those results back to FCS? So this is a really great format. It's had a lot of time to grow, and these files kind of act as the standard arbiter of flow cytometer information that enters into software like FlowJo or the FlowCore libraries in R. Okay, so we talked about several different key concepts. I realized that there was a lot here, and if you've never heard of flow cytometry before, this might have been a little eye-opening and shocking even. But I want you to remember that flow cytometry is imaging for microfluidics in the same way that fluorescence microscopy is about being able to visualize these dyes in a 2D or even 3D field of view. So I hope that's useful to you, and this is all I'm going to say about imaging informatics. So if you have questions about it, I'll be happy to point you to particular papers that might illustrate how to accomplish a certain thing in ImageJ, for example, if the data you're producing could benefit from that. Okay, Well, if there are no questions, I will look forward to seeing you all at 11:30 when we will have our discussion of R continuing. I did add some things to the script online this morning before this lecture, so if you downloaded it last night, you might want to grab it again. One other detail, we're going to try a library in Excel. So down at the very bottom of the script I've uploaded, there's the instructions on how to install a package on your R machine. So if the network is not behaving itself in this room, maybe you could install the Read Excel library on your own before you come into the class at 11:30 today. Great, thank you very much. I'll see you at 11:30.